Hello guys, welcome in the bioinformatics subject. As far as I'm taking structural bioinformatics and chemoinformatics modules for you. Now, in regards of structural bioinformatics, first of all, we need to know what is meant by amino acids, what are the structures of amino acids, how exactly proteins builds up, and what different amino acids contribute to build up specific proteins. Visualize these proteins, we have different web resources with us. Like take example this protein data bank. It is the biggest data bank of protein which is available online for us. It is the structural view of each and every biological components that is built up from the amino acid. This resource is particularly, it is actually powered by Protein Data Bank Archive, or we can call them as the information about the 3D shapes of the protein. Or even more than PDB, it also provides the information about the nucleic acids also, which encodes this protein. Then different types of viral proteins, bacterial proteins, the proteins belongs to mammalian system, invertebrate system, each and everything. Now, who contribute this protein? So the different researchers, laboratory, which are doing different research activities or different the discovery of new different proteins from different organisms. They discover it, they structurally identify them, and they deposit that structure on this platform with a particular accession number. And after deposition, you can see over here on the left hand side these deposition tool is there or tab is there. So once you deposit, it is very important to know that we need a good publication, either it is a national or international with a good impact factor, where the protein, the discovered protein, which is having a good authenticity. Okay, now this tool can be used by different, it is free, completely free for students, different researchers to understand all aspects of biomedicine, agriculture, from protein synthesis to health and disease. As the member of these WWPDB or RCSB PDB, the curates and annotates, or we can, or researcher and student can curate and annotate PDB data. The RCSB PDB builds upon the data by creating tools and resources for research and education in molecular biology, structural biology, and computational biology and beyond. There are various different subjects or fields where these PDB data bank or protein data bank is useful. Okay, we'll see a little bit the information when it is established and who established everything. The protein data bank was established as the first open access digital data resources in all biology and medicine. Today, a leading global resources for experimental data central to scientific discovery. So if I'm working on something specific protein, like just take example, the world, the different laboratories, they are currently working on coronavirus. And so they are working on epitopes and their models and how they interact with the human different surface receptor proteins. They are building these different protein molecules, and they are checking out how exactly they are interacting. So through an internet information, the portal and downloadable data archives, the PDB provides access to 3D structure data for large biological molecules, like take example proteins, DNA and RNA. These are molecules of life. You all know that, and they are found in all the organisms present on planet. Knowing the 3D structure of these biological components, which is very essential to understand the roles of these macromolecules, these biological molecules in human and animal health, or the disease related to different mammalian system or any other animal or organism. Their functions in the plant, in the food, energy production, and its importance to the other topics related to global prosperity and sustainability. So, to understand these biomolecules in the 3D way, or how exactly they respond structurally to in the different macromolecular way, which is very important. The enormous wealth of 3D structure data stored in the PDB has underpinned significant advances in our understanding of protein architecture, culminating in the recent breakthrough in protein structure prediction accelerated by artificial intelligence approaches and deep on machine learning methods. If you see that there is another database, guys. Now other database provides us these web resource that is called it as NCBI. It provides us a window to PubMed, the publication media, or this is the library. It's the National Library of Medicine who provides us different research articles, over 33 million citations for this literature from the Medline or from the different life sciences journal, online books which is available online, sometimes freely and sometimes they are paid. It depends on that journal on in which these research has been published. So PubMed is basically, it's a free resource available to us. It supports the search and retrieval of biomedical and life sciences literature with the aim to improving the health, both the globally and personally. To improving the health means what? This is the ultimate of our research. How we are improving our health, how we are designing different life sustaining different types of drugs, or how we are coping up with the environment, with the nature, everything. The PubMed database contains more than 33 million citations abstract of biomedical literature. Generally, it does not include full text journal articles. It provides us the abstract basically. Sometimes they provide some essential data, some graphical representation or pictorial information, might be it's freely available. Like I said, some publications take charges if you are referring their research articles, and sometimes some research journals are completely free to each and every researcher or the person who is actually looking for it. However, links to the full text are open present when available from other resources. So this PMC or PubMed Central, it is available publicly online since 1996. See guys, when we have started our human genome project from 88, the actual health science department or actual human health research or medical research, it is like started with something new ideas, because up till now we actually at that time there was not present ample amounts of information about DNA, how they encodes, which proteins are there, what different proteins are there, and how they interact with each other. After finding these different theories, different sequences, we are able to what do we call synthesize chemically or synthesize these in by in silico method the different protein structures, and from this protein structure we can depict the function of these protein structures. Otherwise, to isolate that protein, purify that protein, and then check the activity, it's very laborious procedure if you work in the molecular biology lab. There are lots of what do we call, the time consuming procedure is also there. Lots of chemicals we need which are very expensive. So apart from this all research, if we do these proteins, if we construct this protein model in the computer or with the help of computer, with the help of different algorithms, different simulators, definitely we can judge the function of protein and we can design necessary entity like drugs, vaccine, different antibiotic components, everything. So this PubMed is like that, very important. There are books are also available through this PubMed. The research journals are available. The notes are available. Different types of data are there, like reports or short reports, short publication, short communications are there. This is the one type of publication related to biomedical, health, and life sciences. Now, in the protein data bank, protein is actually visualized with the both way, the sequential, different types of formats are there, but the one format which is universally accepted format and which is accepted or which is acceptable to each and every software is the PDB format, that is protein data bank. P for protein, D for data, and B for bank. So this PDB file format is very important. It's the textual file format, describing three-dimensional structure of molecules held in protein data bank. The PDB format, accordingly, it provides for description and annotation of protein and nucleic acid structure, including atomic coordinates, secondary structure assignments, as well as atomic connectivity, their energy, everything. In additional metadata, it is stored. The PDB format is a legacy file format. Remember this, it's a universal file format for the protein data bank, which now keep data on biological macromolecules in the newer format, that is MMCIF. Okay. So protein, this format, the PDB format, it is the very important file format. We'll see that what it is exactly, or how exactly we can visualize these different format or this PDB file. There is a one viewer which is offline. The software is also available, and online it is also available. That is called it as Swiss PDB viewer. It is a very good for software to visualize this protein. Okay, and we can visualize protein in terms of different format. We can visualize this protein in different alpha helices, beta sheet, their turns, coils, we can see everything. But before that, before to see all these formats in the software tools, before run all the models, we need to know how many different types of structures are there regarding the protein. I know that since BSc you are understanding proteins, their property, what different types of amino acids are there. So I'm not going to explain you here what is meant by polar and non-polars and what is meant by negative and positive charged amino acids and how they interact with each other and everything. That is very basic. So we'll see the next thing, which is really necessary for our study, how exactly protein secondary structure looks alike. So it is the regularly or regular recurring arrangement in the space of adjacent amino acid residue in a polypeptide chain. You all know that the chiral carbon is there in the central, right? Then what? The NH2 molecule, or this is the actually we can say that this is the one terminal, the amine terminal. Here there is a R group, you can say. Now what is R group? R group is the actually what do we call it? It is the amino acid group, or it is the uh, uh, adjacent group which decides what that amino acid is. Okay, then the other one is the H atom, and here there is another carbon which is associated with the oxygen, COOH, and then ultimately the other amine group is present. Okay, and like another C alpha, so this is C alpha, remember. So likewise this chain begins. Now how exactly these helices form and beta sheets form, we'll see that in details afterwards, but this is the actual the basic structure of the amino acid sequence and how it joins up. So it maintains by the secondary structure is basically maintained, or these polypeptide chains, they are attached by these polypeptide bonds, you all know that. But the structure, the advanced structure, secondary structure, is actually maintained by hydrogen bonds between amide hydrogen, okay, the amide hydrogen and carbonyl oxygen. Now remember, here the other hydrogen is present, okay. So this is carbonyl oxygen and this is amide hydrogen. So do they form hydrogen bond here only? No. Once they make a turn, like I4. So take example I. I is the first amino acid from where we are beginning. So if you see the to which amino acid, these amino acid, the I amino acid form the hydrogen bond I plus 4. So fourth amino acid having a property so that it can make hydrogen bondings with each other. Okay, so might be it is making a like that turn. Okay, so this is like alpha helices, and the amino acid which is present over here, the amino acid which is present over here, they will present this bond. Okay, hydrogen bonding. Some hydrogen bond is present here, some hydrogen bonds are present here, some hydrogen bond present there. So likewise, and they actually due to hydrogen bonds, they form a structure like that. So they are actually like a peptide backbone. Now how many different types of structure forms, like we can, if we are talking about secondary structure, there are alpha helices are there, then beta sheet structures are there are turns are there, like we can say the bends. Okay, when proteins are forming, the proteins are folding up or they are making bends to each other, and that bend or turns are also called it as the they are also because of these hydrogen bonds. Then there are coils are there, irregular coils are there, we'll see them later. Just remember these four structure comes up in the secondary structure, like I said. So the polypeptide chain is a coiled as a spring. Okay, you all know that. See here how exactly it is coiled. So it is coiled like that way. Okay, it is not in that formation, but it is actually circular, remember. Okay, like that way. And so it forms a bond, or the we in between carbonyl oxygen of one amino acid of the residue of I, forms the hydrogen bond with the amide residue of I plus 4. Okay, remember that concept. Now the alpha helices is stabilized by hydrogen bonds between these I and I plus 4, as per you know. And what is 3.6 residues per turns are present. And these 3.6 residues per ton means how much distance they covers. That means 3.6 residues of amino acids, how much distance they actually consumes, like we can say 5.4 angstrom per turn. So each turn contains 5.4 angstrom. And if you want to know how much angstrom unit actually covers by single residue, we should divide it, like 5.4 divided by 3.6. You will get how much angstrom units per residue, right? The repeating unit of alpha LSS. And these residues are very closely packed due to hydrogen bonds. Okay, now these there are now two types of structures. The one is alpha helical, we say that helical structure, and the other one is beta strand. Now what is that beta strand? There are again two types of beta strand structures are there: anti-parallel beta sheet structure, parallel beta sheet structure. Within this anti-parallel beta sheet structure, you can see here, if the sequence starts from here, and it will coils up and it will again start from here. So the direction is like one sequence is going in that way and one sequencing is going in that way, and there are hydrogen bonds appears in between them, right? These hydrogen bonds likewise. Now this is not helical structure, so no I plus I plus 4 kind of thing is not happening in here in the beta strand, and so this type of thing we can call it as anti-parallel. In the other way, sometimes it happens that some beta sheet structure are not like that way, but some beta strand structure are forming hydrogen bond with these structures. So they are like, the sequence is also going through that way and this sequence is also going through that way. So this is parallel beta sheet structure. Thus a beta strand is fully extended and loosely packed. Now remember, alpha helical is tightly packed, but here in the beta strand or beta sheet structure, it is loosely packed and they can hydrogen bond together to form a beta sheet. Okay. Then next one, that is turns. Now 1/3 of all residues in the globular protein. Suppose the protein is globular, which is having a different shape and size, you all know that, and within that 1/3 component, or we can say 1/3 sequence of that protein is actually contains different types of turns, or it is composed of turns that serves to reserve the directions of polypeptide chain. Turns, yeah, where we were, yeah. So uh, now see guys, turns contains hydrogen bonds between the carbonyl oxygen. Okay, it contains hydrogen bond, and in the carbonyl oxygen of residue I and the amide nitrogen of these I plus 3 amino acid. Now third amino acid from the I. Okay, so I is the alpha amino acid from where you are counting the hydrogen, and you can see not like alpha, it is the I plus 3. These three types of turns, okay, these three types of turns are designated as the first, second and third. Type 1 term occur most frequently because of what, 2 to 3 times more frequently than the type 2. Now the next one is very important turn. See guys, amino acids, the backbone of these amino acids are formed because of dihedral angles of residue. Now what is mean by this dihedral angle? Now remember, each amino acid having a structure which contains two different angles. Okay, two different angle. Now take example. These dihedral angles are there, and these dihedral angles with the backbone which contains two angle, -60, -30 and -90 and 0. Okay, this angle is made by which amino acids? Residue I plus 1 and residue I plus 2. And that forms actually, and when that forms, that turn is, we called it as type 1 turn. Okay, so I'll get back to you when I'll explain this one. See guys, so this is dihedral angle. There are two angles, right? Now see, this is the C alpha, like I said, C alpha. This is the chiral carbon which contains the side group or group chain R group which contains extra hydrogen. Okay, the other carbon atom which is present over here, the carbon which actually attached to the oxygen, so that's why we are calling it as carbonyl carbon. Okay, and these C alpha and N, these bond is actually can turn in this direction, like clockwise direction. Okay, so the bond or the other amino acid, these plane, okay, remember these plane, when this plane is actually turning clockwise direction, it is called it as the phi angle. Okay, the angle which is made by these amide plane with the another amide plane, that angle is called it as phi angle. And when the other bond which is formed from C alpha to carbonyl carbon, this actually angle which is actually turning anti-clockwise way. Okay, anti-clockwise way, and this turn we call it as psi angle. Okay, And these two angles, when they made, these two angles is called it as dihedral angles, guys. Remember. Okay, now we'll get back to our turns. So here, type 1 turn, the residues are making -60, -30 and -90 and 0 angles of the residue of what? These I plus 1, I plus 2 of, and when these type of angles, when they form, that is called it as type 1 turn. The best information is that proline is often found in the position. Proline is always found in the position where the I plus 1 amino acid is there in the type 1 turns, as its phi angle is restricted to -60, means it always turns up to the -60 degree centigrade, degree. Okay. The backbone dihedral angles of residue, if -60, 120 and 80, 0 of residue I plus 1 and I plus 2 respectively, they form type 2 turn. Okay, there are three turns. One turn we saw, the another turn is like that, -60, 120 of these I plus 1, when they form like I plus 1, when I plus 1 is forming -60 and I plus 2 is forming 120, or and I plus 1 is forming 80 and I plus 1 is also forming 0 degree or 0 angle, then they construct type 2 turn. And then when type 2 turn, when we can say type 2 turns, see guys, there are specific amino acids which actually present or occurs on that position at that particular turn, because this amino acid has that property, okay, to form such a kinds of dihedral angles. Okay, so glycine is favored in this position in type 2, as it requires the positive phi value. Okay, next. So based on these, there was a scientist called Ramachandran. He is the Indian scientist. During that time, by understanding each and every amino acid and their compositions, he actually depicted the different amino acids and their attachment and their composition by depicting their mathematical values. Okay, when the one amino acid, X amino acid, attaches to a Y amino acid, and what angle they form based on the interaction of these hydrogen, these oxygen. Okay, because each and every atom has its own electronegativity, their own what do we call, the electron sphere, and when they interact together, they restrict on that position. When they find the stable condition to stay away from each other, to make the distance within them, so they are very stable when they stay on that position. They cannot interact, they cannot share their own electrons with each other. We call it as electronic sphere. That kind of structure also I'll show you. Okay, so these these formation is when it forms, the Ramchandran has studied these things, and he actually plotted on Cartesian coordinate system these different angles, phi and psi angles. So psi angle is on y-axis, phi angle is on x-axis, and from plus 180 degree to minus 180 degree the angle can form, and from minus 180 to plus 80 degree psi angle also can form. So out of that, when he found that the right-handed helix structure is actually formed when the psi angle is in between these range at the negative side, and the phi angle is also at the negative degree. And beta sheet structure, when it forms, when it, just a minute, I'll choose another color, when these angles or these the angles in between these area, when they form the phi and psi, then the amino acid actually form the beta sheet structure. And this plot is very much important for any algorithm to depict the structure of amino acid sequence. Okay, so based on this, he depicted that the part of these. You can see here the part of these area is actually anti-parallel beta sheets. The part of these structures here is the parallel beta sheet. Then different sheets, right twisted beta sheet, left-handed alpha helices comes up over here, and that tells us what is the stability of protein also, is it? That actually shows us the stability of the protein, what exactly do that protein structure actually sustain within that environment or not, what is the energy, what could happen to the protein when we add or when we have different biological components within it. This is what actually happens when we study the Ramachandran plot. We'll see the theoretical information. The G and Ramachandran, he used computer models. Okay guys, he used computer models for that. He used computer models. He used small polypeptides to systematically vary phi and psi. These are the Greek words. Okay, phi and psi, for finding stable conformation. The atoms were treated as a hard sphere. Okay, atoms are treated as a hard sphere. Their dimensions he actually counted or calculated with the help of van der Waals interaction with each other, van der Waal radii. Okay, and then phi and psi angles that causes spheres to collide. Okay, when these two atoms, at what position they can collide and at what position they stabilize themselves. Okay, and these stabilization, this unit, okay, at what conformation they could stabilize. This is the another atom, this is the another atom, so what could be the angle in between them two? Okay, so that's he studied with the help of sterically disallowed conformation. And the next one is what is meant by torsional angle. This is called it as torsional angle. The angle which is forming in referring to other plane surface, in refers to other plane surface, when the one plane tilt around either in negatively making angle or negatively angled or positively. This is what we call as a torsional angle. And how we count? We count it with the help of by plotting a matrices. How he calculated it, that is the best question, right? This is the how exactly he calculated it. He making the a, he plotted the unit of a, y to z axis, then x to z axis, and then x to y axis, and based on that he depicted the d value, that is torsional angle value, which comes in negative way. Okay, the dihedral angle is the angle between two planes. To calculate this angle, you can follow these steps. They calculate the equation for each plane, and it will be the form like ax by cz plus d is equal to 0. And based on this, we can calculate this torsional angle. The torsional angle is cos alpha. Okay, and based on them, based on these angles, we can get the x value, we can get the y value. Okay, this three dimension, and z value. Okay, and when this is the item, this is the atom, and the number of atom is over here, and suppose this is the c alpha of what, the amino acid is glycine atom number, and the their calculation, if we calculate with the help of these dihedral angle, we can get the actual dihedral angle it makes up. Okay, with the other carbon, or this is the carbon atom, how exactly it makes up angle with the other carbon atom. Okay, I hope so you are understanding how exactly to calculate and how they calculate the dihedral angles. So this is the, all these things is a computational equation, but Ramachandran has made it possible on pen and paper. He did it, all these things. So the surface one, so based on the one plane, suppose first plane, how exactly we calculate? The first of all, we predict the three axes with the three different possibilities, distances from apart from different atoms. The equations of plane a, x, b, y, c, z plus d is equal to 0. Based on equation, we got this particular value. Then surface 2, we can get the equation of this surface 2. We will get the equation. If we calculate these both equations and if we depict the dihedral angle with this equation, remember this equation, right? This cos alpha. If we depict the equation, we will get the actual degree and radian. So this is 77 degree if positively tilted, or that means it is the clockwise turned the dihedral angle. And based on this dihedral angle, suppose this is the amino acid has a name 4-MBN. This is the data of that amino acid, and here is the x-axis, y-axis, z-axis, and there is the angle form. You can see here the different angles are there, how exactly these different angles and how they interacted with each other. Okay, I really want to show you these details of how exactly proteins molecule shows up when we take any protein component into our protein simulation or simulator or in the SPDB viewer, how exactly the different amino acid makes angle to other one. Now there are two terms that is really necessary to understand with us, that is scope and cath. So we'll see that in the next lecture.